blood vessels build complex networks throughout the body that span a remarkable total length that could wrap nearly 2.5 times around the Earth. While all these networks are necessary for homeostasis, they can be exploited by various diseases. Introducing tumor angiogenesis, the formation of blood vessels that provides the tumor with a supportive microenvironment rich in oxygen and nutrients required for cancer survival, malignancy, and metastasis. The architecture of tumor vasculature is structurally and functionally chaotic. It's characterized by excessive branching and shunts. This makes it difficult to anticipate tumor progression for prognosis and deliver treatment due to poor perfusion. Existing models struggle with bias from vascular abnormalities, capturing the dynamic patterns in growth rate, and addressing angiogenesis from a multi-level pertinence to the proteinomic, genomic, and pathological sphere. That's why I wanted to investigate angiogenesis, an inherently chaotic network with network science and chaos theory. Chaos theory examines the underlying set of patterns and deterministic laws that are highly sensitive to the initial conditions of a seemingly random dynamical system, like angiogenesis. To this scientist's knowledge, no application of chaos theory has been applied to tumor angiogenesis, despite this being a process so fundamental and influential in cancer evolution and volatility. So I asked, can a computational network reconstruction of tumor angiogenesis mechanisms using coupled morphological, genomic, and physiological parameters, when applied to chaos theory, determine the predictability of tumor angiogenesis? Essentially, I asked, is cancer chaos? First, I established the objectives that the model should achieve, and chose to test my hypothesis on breast cancers due to A, the accessibility of quality mammogram imaging, and B, the BIRADS, which stands for Breast Imaging Reporting and Data System scheme, detailing the likelihood of an image having cancer from a scale of 0 to 6 for accurate labeling purposes. Step 1 was extensive data preprocessing to capture the intrinsic complexity and characteristic dense nodal structures of biological networks while controlling significant variables. I needed research methods to enhance the contrast of the image, segment faint edges, and mask noise so I could binarize and skeletonize the vasculature for network analysis. Step 2 was to acquire the network features and properties to quantify otherwise qualitative topologies of the vasculature. Step 3 was to use exploratory data analysis to uncover general tendencies of biological networks, as well as distinguish angiogenesis from healthy vasculature. First was network feature importance, which was used to understand the extent of causation. Then a matrix of scatter plots between every pair of variables were generated to provide a dataset-wide expansive lens for general correlations. Following this, the 4 most significant variables were plotted in a standard space representation, and lastly, k-means clustering was then used to find the optimal number of clusters and describe the networks on a novel polar coordinate graph to observe how these variables differentiate among the different clusters. Step 4 was to apply chaos theory. The Lorentz attractor equation was implemented to conceptualize tumor vascularization on a 3-axis phase space. Its constants were manipulated to represent the min-max normalization of the 3 most significant network variables. Derivative calculations of the current point using the 4th-order Runge-Kutta integrator were used to estimate the subsequent point of the ordinary differential equation in a time-step fashion. Finally, the Lyapunov characteristic exponents, fractal dimensions, and bifurcation diagrams were used to determine the overall system's measure of chaos. Step 5 was protein targeting and molecular docking with statistical tests. Lastly, in vitro validation with Pfizer polycephalum was conducted to culture the slime mold in adverse, neutral, and generative agar plates to simulate biological network morphogenesis in various tumor microenvironments. Overall, it was found that breast cancer angiogenesis exhibited a higher Lyapunov exponent than in non-tumorous vasculature, confirming the hypothesis that angiogenesis architecture can be modeled by deterministic chaos. It was observed that the number of nodes, the mean mesh size, and the number of segments exhibited the greatest, second greatest, and third greatest significance in a given network's BIRADS label. The general trend of the number of nodes exhibited by the breast tissue vasculatures increase with a greater probability of cancer. Considering that vascular sprouting, the process by which endothelial cells respond to angiogenic clues by expanding their filopodia, the higher number of nodes thus explains the heterogeneous vasculature in addition to advantages such as increased permeability, diffusion, perfusion, and surface area. The number of segments across the birads also appears to have increased and can describe the connectivity of a graph as a scale-free network due to the heavy-tailed power law degree distribution seen. Cancerous networks demonstrate a much greater relative commonness of segments with a high degree of preferential attachment. It is inferred that this is due to the cocktail of pro- and anti-angiogenesis growth factors interplaying between the vasculature, the tumor, and the body's immune response. The mean mesh size rather demonstrates a negative nonlinear relationship. Considering the process by which angiogenic cells connect with each other to form continuous endothelium loops that promote parasites to form capillary walls, a smaller size thus could enable more blood to circulate through adhesion molecules, benefiting tumor survival. Lastly, there was a remarkable correlation between the magnitude of chaos and the likelihood of cancer. Not only were the Lorentz equations striking in themselves, but they visually confirmed the findings from the bifurcation diagram, fractal dimensions, and the set of Lyapunov characteristic exponents. Based on the signs of the Lyapunov exponents, the fractal dimensions of the BIRADS1 network is a stable fixed point, and this can be visualized as all the perturbations are represented as spirals that eventually reach zero. The same can be done for the BIRADS 3 and 4 network, but now demonstrating a more loosely wound and continuous spiral. Then in BIRADS 5, where there's the greatest probability of cancer, we can actually see a positive Lyapunov exponent. So this study provides the first preliminary evidence which suggests that tumor angiogenesis is indeed chaotic, and thus the concepts relating to chaos theory can be applied to underlying chaotic patterns that have yet to be explored in angiogenesis. These applications can range from clinical diagnosis to bridging biochemical and mathematical research for novel drug discovery. And some next steps for this project include further validating the model, converting the code base into an open source library, and integrating the computational algorithm into an innovative imaging hardware. Ultimately, the fascinating network analysis of chaos theory and its affinity to angiogenesis compel for further investigation, countless applications in predicting tumors' vasculature, drug discovery, and uncovering the chaotic rules that drive biological networks in and around us.